When we were in lecture last time, we were talking about different ways in which substances can be moved across a cell membrane. We talked about diffusion as one of the options. Remember that refers to the passive movement of substances from an area of greater concentration to an area of lesser concentration. We also talked about diffusion, which was or I should say osmosis, which was the diffusion of water across a semi-permeable membrane, where you have some dissolved solute that's trapped behind a semi-permeable membrane through which it cannot pass, but water can. And due to the nature of how diffusion works and the laws that regulate it, water is drawn across the membrane toward the solute. And then we talked about different mediated transport mechanisms, including this facilitated diffusion, which was a carrier protein is going to be involved in helping substances move across the membrane from an area of higher concentration to an area of lower concentration. And then we talked about active transport, where in this case a carrier protein is involved, but rather than passively assisting a molecule move across a membrane, this is going to require ATP energy. And this is going to generally move substances in the opposite direction, going from an area of lower concentration to an area of higher concentration. So it works against the concentration gradient, or against the natural direction of diffusion. And there are a couple of different types of active transport. There's secondary active transport, which I'll describe in just a moment here, in which molecules can be moved in the same direction or moved in opposite directions. Now on the screen you'll see an example of active transport mechanism. This is something called a sodium-potassium pump. This is the one that I described on the board last Friday, where you have a carrier protein that's embedded within the membrane, and ATP is going to be used to power this particular little pump protein. And this one moves 3 sodiums across the membrane and 2 potassiums across the membrane, but they go in opposite directions. So 3 sodiums get moved in, 2 sodiums get moved out. And this is a mechanism that the cell uses to concentrate sodium outside the cell and to concentrate potassium inside of the cell. Here you see another example of secondary active transport. Notice the pink protein on the right. This protein is going to do 2 things that are kind of interesting. One is that it's going to passively allow sodium to move through it according to its concentration gradient. So imagine that in this cell, the upper part of the image represents the outside or extracellular space. The lower part of the image represents the intracellular space. And because of the action of the sodium-potassium pump, the sodium is concentrated outside the cell. So it wants to diffuse into the cell. And the pink pump allows that to happen. It allows sodium to passively move through it to get onto the inside of the cell according to its normal concentration or concentration gradient. But while that sodium is diffusing through that pink protein, that movement of sodium across that pink protein provides energy to the pump, allowing it to move glucose across the protein or across the membrane. Now here's kind of a way of thinking about this. I often think about these secondary transport molecules as being kind of like a revolving door, like you see at a large hotel or something like that. And sort of imagine this scenario where you have a small child who is too weak to actually cause the door to revolve. And so when they get into the door, they can't push it, and so consequently it won't turn, and they can't get into the building. But if they wait until a big person comes along and hops into the revolving door, and the big person pushes the door and starts it spinning, the little kid can actually hop in and take a ride through the revolving door and get into the building. That's similar to what's happening here, where the passive movement of the sodium across the membrane kind of gets the door spinning, and glucose kind of hops a ride once that protein begins to work. This is an example of secondary active transport. Finally, there are a couple of other ways in which we can move substances across the membrane. The two are called endocytosis and exocytosis. Endocytosis is a mechanism in which a cell can take large particles and pull them in. And this particular image kind of shows a summary of what's going to happen. The cell makes a sort of pit, a pit probably is the best way to describe it, where a portion of the membrane begins to kind of suck inward, forming an indentation or a depression in the cell membrane. The substance is kind of gathered into that depression, and then that depression sinks deeper and deeper into the cell until it finally pinches off, forming a vesicle or vacuole deep inside of the interior of the cell. So let me show you some examples of how this might work. So again, it's for internalization of substances, and we can kind of go through a couple of different types. You sometimes hear endocytosis referred to as phagocytosis if it's involved a cell that's kind of consuming something else. Let me show you an example on this image here. Okay, what you are looking at, let's focus primarily on the image on the right. You see the cell membrane shown by the double blue lines. The interior of the cell is shown as yellow. And notice we've got these purple molecules outside here that the cell wants to kind of move into the interior. So notice that a small pit begins to form, and these molecules get picked up by these specific proteins that hold them in place. The pit deepens, and eventually you can imagine that the pit kind of sinks in, and the two parts of the cell membrane kind of adhere to one another as this vesicle pinches off and goes inside. It probably resembles something that looks a little bit like the formation of little bubbles or beads in a lava lamp, where it kind of things pinch off and the rest of it kind of heals over. And notice now at this point that you have a vesicle inside of the cell that contains the molecule that you wanted to bring inside the cell. And frequently after this has occurred, the cell releases an enzyme that's going to degrade the membrane surrounding that molecule. It will kind of break it apart, releasing the molecule into the interior of the cell. Here's again another sequence of cartoon images showing the same kind of idea, where you can see more clearly how the forming pit kind of gathers the molecule in, encloses it, the membrane kind of seals over, and then the vesicle is brought inside. Now the opposite of this can also occur, in which a cell can form a vesicle out of phospholipid bilayers, fill the vesicle with some kind of substance, and then push that vesicle toward the membrane. And as the vesicle touches the membrane, it becomes part of the membrane, and then the membrane opens up, releasing the molecule to the outside. That process is known as exocytosis. So when you bring a substance into the cell, we call it endocytosis. And when you take a substance and put it into a vesicle and use that as a way of releasing the substance outside of the cell, that's called exocytosis. So here we can kind of see in a series of these images kind of taken separately, you can kind of imagine an animation of sorts in which the substance is kind of gathered into a pit. The pit deepens and begins to close off, and now you've got a vesicle on the inside of the cell. The opposite would happen, where the cell builds a vesicle and fills it with some substance, pushes the vesicle toward the membrane, and then the vesicle membrane becomes incorporated into the cell membrane, and the substance is then released outside of the cell. So that's an example of exocytosis, or at least kind of explains the process by which exocytosis occurs. Here you can see an actual example of what the cell might do with that. Let's suppose that one of the jobs of this cell is to produce some kind of hormone that it secretes into the environment around the cell. The hormone would be produced by an organelle within the cell known as the Golgi apparatus. The Golgi apparatus builds the molecule and then forms it inside of a vesicle, and then pushes the vesicle toward the cell membrane, and then the vesicle fuses with the membrane. Vesicle opens up, and the substance that was inside of the vesicle is now released outside of the cell. So that's an example of how the cell might use a particular mechanism like this. And so this is how, for example, digestive enzymes are secreted by the pancreas, how mucus is secreted from salivary glands, and even the formation of milk, that should say milk, not mild, by mammary glands.